West Africa, and Southern Africa has 4 hypotheses, none of which have been proven. You mentioned 2 of them. One is genetic differences between the different geographic locales. The second is the circumcision. The third is the fact that HIV-1C emerged in Southern Africa rather recently, and that has not emerged in Northern Africa. And the fourth is the concordance business about the behavior, which is a little leaky as well. There's leaks in every one of those hypotheses. None of them have been rigorously tested. They're all out there. There are proponents for each one. My escape answer is it's probably a little of each. But let me tell you a little bit about circumcision. Circumcision has a relative risk of 2, which sounds like a lot, but it probably won't explain the difference between a 25% prevalence in Botswana and Namibia and a 1% prevalence in Nigeria. It just doesn't, that dog won't hunt, okay? It does a little bit, but not all of it. The concordance hypothesis, I've heard different. I'd be interested in your opinion on that one, because although I was impressed by the book about it, I have found that my colleagues who studied over there say, well, the behavior isn't that much different in the different parts of Africa, as was explained. With respect to the genetic differences, there are genetic differences, but they're less than the differences between Africans and Europeans and Africans and Caucasians, and I explained to you that's pretty small anyway. So I doubt that would explain it all anyway. The one that I kind of like that probably explains at least as much as the other 3 is the HIV-C emergence. We're starting to see differences in pathogenesis associated with HIV-1, the different clades, and we're getting better at measuring it, and perhaps it's the interaction of that and the genetic predisposition in other kinds of the country. But the truth is, before the early 90s, HIV-C didn't exist in Africa, and a lot of the places, including Southern Africa, was B. And at that time, the frequency was really low down there, like 1 or 2%. So almost within a 5-year interval, HIV-C took over and the prevalence went way up. And the only B you see in Southern Africa now is among the gay men. Now, you go figure. I don't know exactly what it means, but it sounds to me like there's something about the transmissibility of HIV-C and the pathogenesis that we need to understand better.
Maybe in combination with immunogenetics.
Absolutely.
Now, what about Western Africa and the United States?
Oh, the second part of your question, yes. The genetic footprints associated with migrating out of Africa that we see in African Americans are at best only softly understood. We know that African Americans have a distribution of Caucasian ethnicity because of the gene flow come in from Caucasians, males mostly, since the since the slaves were brought to North and South America. What we don't really understand very well is the signatures of different geographic locales. Although you can go on websites and buy, for a small amount of money, a description of your ethnicity, those of us who study the differences between ethnicities carefully pretty much know that determining ethnicity is not just a matter of looking at a half a dozen or 50 markers. You really need to take thousands that have been specifically selected, and that hasn't been done really that carefully for any individual or others. When I say that to New York Times writers, I often get a phone call from some lawyer telling me they're going to sue me for ruining their business. But I will say that I think I can defend what I don't know about that particular approach. And so the answer to your question is, we would like to understand it a little bit better, and I think it might be possible if we had a better understanding of the dispersal of type-specific variation in different geographic locales in Africa. We don't have that yet. When we get that, then we can go back and revisit this question. It's an important question. I think it's solvable, but we don't have the tools yet because we don't have it quite investigated that well. I will say something about the Pacific Islanders too, and I hate to bring this up, but in the places that I visited, like in Hawaii and in the Pacific, the sensitivities of the leaders of many of these ethnicities, not the people themselves, but the leaders of these ethnicities, is huge. They don't like to be fiddled around by geneticists trying to figure out what it is that makes them more susceptible to this or that. And so the numbers that are required for understanding the kinds of heterogeneities you were bringing up ain't ever going to be understood unless we can change that resistance.
Interesting.
I'd just like to second what Gail said and say that I think that her comments about 13 to 15-year-old black MSMs in the United States apply with equal force to girls in sub-Saharan Africa and in the Caribbean. Barbados has an explosive epidemic in young girls that age, and that's a country with 100% literacy, 3 national newspapers, and a comprehensive English-style public school system. So when the category is 13 to 29, I think it has the effect of disguising an epidemic in young teenagers, and that has implications for sex education programs, for prevention programs, and so forth. And I'd like to see that category at least broken into thirds. So we're talking about really young people at the beginning of their sexual life. Well, in Barbados it's more like 11, but you know, they are different. And what you're missing is an opportunity to step in and also to convince people that sex education is necessary at an early age, because 29 is pretty grown up. I actually remember 29.
I just want to say I agree with you. The only reason why for the 06 data we've had to keep it for 30 to 29 is we just don't have the power to unpack it. But over subsequent years, as we begin to amass more incidence data, we'll be able to do that.
A question for the two of you combined. The presenters, both of which talks were wonderful. How many years away are we from being able to model incidence data that will be the combination of plague type, genetic susceptibilities, and other more social factors? And then I have a second question. Kevin mentioned today about the push for testing being linked to the 50% immediate reduction in transmission. But it was especially disturbing to see the articles from the UK in the summer showing that going away. And do we have any data for the United States or any surveillance system among HIV positive persons in the United States to say whether we're seeing, especially among MSM, higher rates of unprotected sex among HIV positives?
Okay, do you want to?
You go ahead.
So I think what you've mentioned with that mathematical model would be the holy grail. Where we are now at CDC is to develop a resource allocation model which uses a transmission dynamic model for the US for HIV transmission, and then incorporates into that as inputs what we know about the effectiveness of various prevention interventions. So we're able to say, with this combination of interventions, what is the likely impact on the HIV trajectory? And so that's going to be really exciting for us to help us. But I think it's still a few years off to incorporating some of the genetic information. The second part of your question was around one of the hypotheses on the effectiveness of HIV testing as a prevention intervention is the belief that those who test positive will reduce their risk behaviors. And you're right, we are seeing very different pictures across the various transmission groups. Certainly among many heterosexual populations, we see tremendous declines in risk behaviors post-diagnoses. Across various Western industrialized countries from MSM, we see very different pictures. It just goes to show the importance of secondary prevention with people who are diagnosed positive, that part of our prevention strategies is not only keeping negative people negative, but those who are positive to ensure that we're working with them to reduce their risk of onward transmission. And it's going to be a lifelong, we need to change the frame in which we're doing this prevention work. In the same way a diabetic, you're going to be seeing your doctor for years, perhaps for the rest of your life. If you're HIV positive, you're going to be seeing your doctor for the rest of your life. Where are the conversations around prevention? Where are the conversations around, have you had a new sexual partner? Are you changing your relationships? Have you had an STD? Where are those conversations happening with HIV positive people? And that's the sort of thing that we need to get to. That's the place we need to get to.
Are we seeing rising sexual behavior in the U.S.? Do we have data to confirm that among positive people?
I can't think of any now. The good news is that the transmission rate for HIV in the US is declining. So if you think about the number of new infections which are occurring per 100 HIV positive people, that has declined from about 7 in 2002. It's now about 5.3. So overall, we know that either as a result of antiretroviral therapies or individual level behavioral change or whatever combination of prevention interventions, transmission rate is decreasing in the US, and that's another good sign.
We're going to take two last questions, one here and one up there. Please.
You probably hear this more and more all the time after presenting this data publicly since August, but as someone who's been around in the HIV research and clinician, and also the gay community to watch the rates drop, as you showed so nicely from 83, that occurred because of the gay community saying that we're going to stop this epidemic because we have to. It's affecting us. But that really had very little or any governmental influence involved in it. It was very successful, as you showed, along with the fact that since people were dying so fast, people responded because of the fear of HIV. Could you describe for us what the National HIV Plan or strategy would look like to respond to this? Because what you're showing here is a failure of that plan working. Among men who have sex with men, whether they're gay or not, and among young men of color, that plan has really failed. That the ABC program in a society where having gay sex is not even legitimized as a part of sexual education. How is the CDC going to address that?
So thank you for that question. I was hoping somebody would ask that. So the first thing is that there is no national plan for HIV in the US. So that really answers the first part of your question. We have been operating for the last 27 years without an overarching framework for HIV, which includes not only prevention, but treatment, care, policies which need to be put in place to tackle human rights and just social justice. All those things which should be components of a national plan have not been in place in the US. So one could argue that perhaps it's unsurprising that we find ourselves at a crossroads now of saying, actually, we've been doing this for 27 years, we have some successes, but we should be doing a lot better. There is a prevention strategy that CDC has been in charge of, which we produced in the 90s, we've updated in 2007, and we're working on to 2020. But what we need is a comprehensive approach, because we need an approach that will get NIH, CDC, HRSA, CMS around the table to say, what do we need to put in place? The Department of Justice needs to be at the table because of the incarceration issue, which I mentioned. Why are we sentencing young black men to years in prison for having an ounce of marijuana? It makes no sense. And those are the things that need to change if we're really going to be getting ahead of the curve. So one of the candidates has said that they support a national HIV plan. The other candidate has looked at more community level and individual level approaches to HIV prevention. We were on the Hill yesterday for a congressional hearing on HIV prevention, and the calls for a national plan have increased tremendously. And I'm hopeful. I'm hopeful that whatever happens post-November 4, whatever happens post-November 4, we will be moving into a new era with these new data on HIV.
Do you think that CDC will ever be able to talk about sex?
I've just spoken about sex for half an hour. Of course. And, will we be able to, let's speak afterwards. I think we will be able to speak about sex. I think, as with all, I've worked in the Caribbean, I've worked in Europe, I'm now working in the US. The role of the public health practitioner is to look at ways in which you navigate the minefields of politics to get the job done. And sometimes it means that we have the conversations, but at a different decibel level, because the context is not where it needs to be. But that doesn't mean that we don't have the conversation, and we have continued to have the conversation as well.
I'd just like to agree with Kevin and the tenor of this audience and just make one minor comment, which is that as a geneticist looking at this, what I see is a tool for identifying new ways to treat this disease. I don't see the solution coming out of genetics for understanding all the differences. I see it coming out of these environmental factors, these behavioral factors, and health education. Right now we have things that work, such as health education, work very well. And probing around and waiting for the geneticists to come up with, genetic explanations is nonsense. I mean, we have effective things that can work right now. The geneticists are trying to find these things because we'd like to treat the patients who are already infected with natural products that basically mimic natural evolved genetic defenses. So we're more in the discovery phase for trying to help rather than trying to explain the epidemiology. I really think the explained fraction associated with all the genetic factors will be lucky to hit 20%.
Last question to the patient gentleman in the back.
Question regarding your statistics for the Latino/Hispanic population. Does that include the immigrant population or people that are self-identified
Good
as
question.
either Latino or Hispanic? Because I feel that when doing research, you clump them into Hispanic, but then there's people who self-identify as Latino, so then you automatically push aside people. And also, are these intervention measures going to include the immigrant population in regards to sex education and all those other interventions?
Great, thank you for your question. So yes, it includes people who self-identify as Hispanic and Latinos. I thought the question you were going to ask me is whether it included data from Puerto Rico, which has been one of the things that we've been drawn over the coals for recently, in part because the incidence data presented was for the continental US. We don't have the incidence data yet for the US territories, but when we do, we'll be able to incorporate them to have a much more comprehensive picture. And one of the reasons why that's important, especially to Hispanics and Latinos, is it will increase the proportion of infections which are attributable to Hispanics and Latinos, and that will be an important advocacy tool. So CDC is committed to doing that. The second part of your question on illegal immigration or immigrants and how they're incorporated into the surveillance data and our prevention approaches. We have gone on record as saying, as a public health agency, the most important thing for us is to prevent these diseases, irrespective of migration status or legal status within the United States. And we've made it quite clear to states who we fund that there should be no discrimination on the use of our resources based upon immigration status. So as far as our surveillance, prevention programming, et cetera, is concerned, it's really an open door policy, because at the end of the day, we're dealing with an infectious disease, and it would be foolhardy for us to do any sort of discrimination based on that.
Thank you. I'd like to thank Dr. Fenton, Wyatt, and O'Brien.
Thank you.
Lunch will be
Fantastic
served
talk.
in the
Really was.
DSRB.
Wonderful.
And you're so passionate about it. I know you are, but it was just great to see you both work. Do they know where lunch is?
It is. I know after lunch, everybody kinds of run away. But it is a great pleasure for me to introduce Dr. Irvin Chen from the Departments of Microbiology and Immunology and Medicine. Dr. Chen has a PhD in molecular biology, and he has an extensive history in leading multidisciplinary teams and has developed and produced a number of highly significant contributions in HIV research. For the past 16 years, Dr. Chen has been the director of the CFAR, the Center for AIDS Research here at UCLA, and is the founding and current director of the UCLA AIDS Institute. Under his leadership, CFAR has been the mainstay of AIDS research on the campus for its more than 200 investigators and faculty. It is one of the largest, most diverse, and productive CFARs in the nation. With over 200 publications to date, Dr. Chen has made a number of noteworthy scientific contributions to retrovirology. Among these, he has been the first to molecularly clone a complete human T-cell leukemia virus. He has also been among the first to characterize infections of the brains of patients with AIDS dementia. So instead of reading his very impressive CV, I would like to just say, Dr. Chen continues to transform the face of AIDS research with his novel scientific discoveries and observations, and it would be impossible for me to give you a summary of them all. So please welcome Dr. Chen.
Thank you very much, John. I also am happy to see everybody back from lunch. And I know many of you have heard me speak before, so I appreciate you coming back to hear me one more time. What I'd like to do today is follow up on what we heard this morning, particularly from Dr. O'Brien, who told us about HIV-1 susceptibility and resistance genes. And the very first one that he described back in the early 1990s was a mutation of CCR5 called CCR5 delta 32 that rendered individuals having this allele, homozygous, that were homozygous for this gene, resistant to HIV-1 infection. And heterozygous individuals had slower progression to AIDS. And you heard this morning that Dr. O'Brien is continuing to characterize a number of other genes that might also be involved in HIV-1 infection and AIDS progression. And maybe someday, not yet, but maybe someday we're going to be able to put all of these genes together and actually be able to manipulate the human genome in a way that can render individuals resistant to the effects of infection by HIV-1, and those that are already infected, make them resistant to further progression by the virus. We're not at that point yet, but what I'd like to tell you today is some of our infant studies with CCR5 in trying to ablate CCR5 artificially rather than in the natural form that Dr. O'Brien described, artificially ablate CCR5 and then examine the effects of that ablation on HIV-1 infection and just the effect of the ablation itself on host animals. So what I'm going to go through briefly some published work and then go into some studies that have not yet been published yet. And when I finish, I want to tell you, because I don't want you to think that I'm going to show that we can cure AIDS by knocking out CCR5. Not yet. So I'm going to caution you that we are working in that direction, but I'm going to take you through the various trials and tribulations of getting there, and it hasn't been easy. So let me go to the first slide. Okay. This is just a very simple summary of what Dr. O'Brien presented this morning. As I said, individuals that are delta 32 homozygous are apparently normal, except in some individuals that appear to have a greater susceptibility to West Nile virus infection. There is no CCR5 expression in individuals that are resistant to HIV-1 infection, except for a few cases of individuals that were infected with an X4 tropic strain of HIV-1. Importantly, individuals that are heterozygous for CCR5 delta 32, they have reduced CCR5 expression and they have slower progression to AIDS. So if we can either ablate completely or reduce the levels of CCR5, given this data, it should be possible then to either prevent infection or slow down the progression of HIV-1 infection to AIDS. And the way in which we want to do this, let's see, is there a pointer? This one? Oh, it's too hard, I can't. Okay, I'll just try to. The way in which we chose to do this was with the discovery of RNA interference. RNA interference was a process that was first described in plants and in worms that can ablate specific genes. And in plants in particular, where it's been investigated extensively, it's actually serves an antiviral function against plant viruses. And so it makes sense then that we might be able to use this to prevent infection by HIV-1. And so the very simple approach is just to see whether we can knock out CCR5 and prevent HIV-1 infection. And so this slide here shows the overall scheme, what we hope to accomplish, and that's to take an siRNA in the orange here, directed against CCR5, messenger RNA in the blue. Once that RNA binds to the target RNA, it leads to cleavage of the RNA. The RNA can no longer produce CCR5 protein, and thereby HIV-1 will not be able to infect cells. So that, in a nutshell, is what we're hoping to accomplish. But getting to that point has not been trivial, and I'll show you why. This is the vector we use for delivery of the shRNA into cells. The RNA itself is produced as a hairpin, which is then cleaved by an enzyme known as dicer, and then the actual functional siRNA is a double-stranded RNA molecule that is homology to CCR5 RNA, and that leads to cleavage of CCR5. So we introduce this into cells actually using HIV-1 itself as a vector. These are studies published several years ago showing that when we do introduce this into T cells, it is effective. We can knock down CCR5. And what we're looking at here is flow cytometry. So the cells on the left are cells that are not transduced with the vector. Cells on the right are transduced. And if you compare, for example, this population, which are the cells that have the CCR5 siRNA, with this population, which has an siRNA against an irrelevant siRNA, you can see that CCR5 levels are reduced in this population. So it does work. We can knock down CCR5 with an siRNA directed against CCR5. And the next question is whether we can prevent infection of these cells by HIV-1. And here now we use a virus that's tagged with a reporter gene, heat-stable antigen, murine heat-stable antigen, and that reporter gene allows us then to visualize whether a cell is being infected by HIV-1 or not. And it's a lot easier if you just look at the numbers here, but the percentage of cells that are infected here is 2% versus 11% in the control cells, and so we are getting a reduction of infection by HIV-1 within these T cells. And another way of looking at this is if you look at HIV-1 P24 production in the supernatant of the cells, so that those cells that have the CCR5 shRNA are producing less HIV-1 than the cells that are transduced with control shRNAs. So this is all well and good, but then it turned out that we ran into our first complication, which is that the cells that are transduced with the shRNA vector showed toxicity. That is, they couldn't be maintained over time. And this is just one experiment here, but over a 15-day period of time, what you can see is that the cells that harbored the shRNA began to decline over time relative to control cells, and as I'll come back to later, relative to cells that are expressing the shRNA using a different promoter. So there was something about the expression of the shRNA that was causing some toxicity within the cells. And so we had to figure out first, before we can put this into people, we had to figure out first what was causing this toxicity. And one of the things we noticed was that if we express the shRNA using different promoters, we can generate different levels of shRNA. So we hypothesized that perhaps it was the levels of shRNA that were different. And if we express shRNA using a U6 promoter, these are polymerase III promoters. U6 promoter, we get this level of shRNA. Using an H1 promoter, we'll get this level, much less shRNA. And then if we then look and see what the effects of these expression of these shRNAs in cells, whoops, sorry. Go back to here again. Then we can see that if we express here with an H1 promoter, we see much less, if at all, any toxicity compared to that expressed from the U6 promoter. So it appears then that high-level expression of the shRNA is not good for the cells that are being transduced. So we had to try to figure out a way to get lower expression of shRNA but still maintain its potency. And this slide here just shows that part of the reasons for the toxicity is a result of apoptosis in the cells. We're looking at a marker on the surface of the cells that's a measure of apoptosis. And one thing to note here is that, first thing to note is that those shRNAs that are expressed by the U6 promoter generally have higher levels of apoptosis than those expressed by the H1 promoter. The second thing to note is that it's not specific to any specific shRNA. So shRNAs directed against CCR5 here, or shRNAs directed against luciferase or LAG-Z, which have nothing to do with CCR5 or HIV-1 infection, also show elevated levels of apoptosis. So it seems to be a general property of some but not all shRNAs that they're toxic to cells, and part of the effect of that part of the reason for that toxicity is apoptosis. So as I told you earlier, we can express these shRNAs with H1 promoter, get less expression of shRNA, and looking at two different shRNAs here, we can show you that by expression with the H1 promoter, we get less toxicity within cells. Okay, that's great. But the problem was the following slide, that if we express using the U6 promoter, we get good downregulation of CCR5. CCR5 levels in the transduced population here are much lower than the non-transduced population. But if we express using the H1 promoter, the promoter that expresses less shRNA, apparently it's not sufficient to downregulate CCR5 very effectively. I mean, there is a small amount of downregulation, but it's certainly not as effective as we downregulate using the U6 promoter. So we had to find a way that we could express low levels of shRNA while at the same time maintain the potency of the shRNA. And the simplest thing to do was just to try different shRNAs. So we screened a number of shRNAs, and unfortunately, we screened about 8 or 9 shRNAs, and none of the shRNAs we screened, they all worked with the U6 promoter, but when we went to the H1 promoter, expressed lower amounts of it, it just didn't work anymore for downregulation of CCR5. So then Danson Anne, who was in the laboratory at the time, decided to take a brute force approach. Since we weren't able to do it by using predicted algorithms, you took a brute force approach and synthesized essentially 2,000 different shRNAs. So the CCR5 gene is about 1,000 nucleotides in length, the coding region at least, and if you consider making shRNAs in the forward direction, reverse direction, in theory you should have about 2,000 shRNAs, and he started screening them through these shRNAs. And fortunately, was able to find one shRNA that was able to downregulate CCR5 quite effectively using the H1 promoter. And here it is, this one here. So the transduced population, you can see, has reduced levels of CCR5. And compared with the earlier shRNAs I showed you expressed from the H1 promoter, which have much reduced levels of, not as effective as reducing CCR5 levels, this one worked quite well. And showing that it also works, that last experiment was done in T-cell lines. This experiment here is showing that it works in primary T cells. Here, primary cells that are transduced show good downregulation of CCR5. And then finally, and most importantly, was to ask whether this novel shRNA that we identified, is it toxic to cells? And the bottom line is that it is not toxic to cells, or has much reduced toxicity to cells here. So again, here is a U6 expressed CCR5 shRNAs. You can see the downward decline of transduced cells over time, whereas the H1 expressed CCR5 is relatively stable over time compared to that of a vector without any shRNA. So it appeared now that we had now an shRNA that could be expressed by the H1 promoter, so lower levels of expression, but still maintain potency and was not cytotoxic to the cells. So now we're at a point where we wanted to be able to test this in a setting where we can maintain stable expression of the shRNA, because ultimately there's no real advantage to using shRNA over conventional chemical therapies for HIV-1, except for the fact that by using vectors, stably expressing vectors, you can maintain it, hopefully, in an individual for their lifetime. And so the ideal situation would be to put this into a person, a one-shot, have it expressed, and prevent infection of the cells by HIV-1. And in order to do that, we have to put it into a particular cell type that will be maintained over the lifetime of an individual. That won't work if we put it into T cells, as I've been showing you so far. T cells have a limited lifespan. So we need to put it into a cell that is going to maintain for the lifetime of an individual. And that type of cell would be a stem cell. So the first set of experiments I'm going to show you here are in vitro studies looking at modeling of shRNA transduction and expression in stem cells in vitro. And the second set of experiments I'll show you will be modeling it in a monkey model system. So these studies here, the first studies, because we're to look in macrophages, because macrophages can be easily differentiated from CD34 positive progenitor blood progenitor cells in vitro using the appropriate combination of cytokines. And then you can see whether if you transduce a CCR5 shRNA into the stem cell, convert that stem cell into macrophages, then will you get downregulation of CCR5 in the macrophages? And we were concerned about, again, we wanted to make sure that we can maintain that shRNA within cells, given the fact that we were seeing toxic effects of the shRNAs over time. Even though in the T cells we weren't seeing the problems with the new H1 expressed CCR5, but we still had to prove to ourselves that by transducing a progenitor cell and converting that progenitor cell into a macrophage, that the shRNA could still be maintained and downregulate CCR5. So this is the experiment. It was done, and in order to make it easier for us to analyze the data, we introduced into the cells two vectors. One is, one vector, push the wrong button here. One vector is a target vector. This vector contains a reporter gene known as mCherry that fluoresces red, and that mCherry is fused with the target sequence for the shRNA for CCR5. So it's the specific 20 nucleotide target sequence that the shRNA recognizes. So if the shRNA works, it's going to cleave that target sequence and then eliminate the red color from the cells. Okay, so it makes it a very easy indicator for us to see if it's working. The second vector is the shRNA itself, expressed here. And both of these vectors were then transduced into the CD34 positive progenitor cells. The progenitor cells then differentiate into macrophages, and we ask, are we seeing shutdown of the CCR5 reporter? And so again, if we see if it works, if it's downregulating the target sequence, we will not see red color. And the presence of green color is the presence of the shRNA vector itself. Okay, so first of all, up here, this is just a target sequence itself. We put the target sequence in, the macrophages fluoresce red. If we put the vector sequence alone, this vector sequence here, the macrophages fluoresce green, but do not show any red. If we put an shRNA against luciferase, an irrelevant shRNA, there's nothing to do with CCR5, we see the vector, we also see the target sequence still being expressed. But if we put in the specific shRNA against CCR5 and the mCherry with the target sequence, now we can see the cells are transduced with the shRNA vector, but the target sequence, target vector is ablated, meaning that the shRNA is working in those cells on that specific target to prevent the expression of the red mCherry. So we did this because it's an easy way to look at the activity of the shRNA, but we also want to look directly at CCR5 itself expressed in the macrophages. And to do that, we used real-time PCR to measure CCR5 mRNA levels within the macrophages. And here again, you can see that using an shRNA against luciferase, we have this level of CCR5 mRNA. If we put in the CCR5 shRNA, now we get downregulation of CCR5 mRNA. So we know now that we can introduce the shRNA into CD34 positive blood progenitor cells, differentiate those cells into macrophages, and those differentiated macrophages will have downregulated CCR5. We can't do the same thing in tissue culture with T cells because T cells require either animal model systems. There is no easy way to do in vitro differentiation with T cells. So to look at the other lineages of cells, we went to an animal model system. And this animal model system was done in collaboration with Robert Donahue at the National Heart, Lung, and Blood Institute at NIH. And he has a great facility there for doing animal transplants. And basically, it's like doing a bone marrow transplant in humans. He takes the monkeys, he mobilizes their CD34 positive cells from the bone marrow using stem cell factor and GCSF, isolates the CD34 positive cells, which we then transduce with our vectors, and then put them back into irradiated animals. And then those animals will reconstitute all of the hematopoietic lineages from the CD34 positive cells that are given to the animals. So let me just show you. As with all the experiments here, there's always complications. The main complication here was that when we analyzed the CCR5 sequence, it turned out that the target sequence differed from the human target sequence by a single nucleotide. Okay, and in RNA interference, a single nucleotide can have a very dramatic effect on the potency of the shRNA. And so what we had to do first, we had to create a new shRNA that recognized the rhesus CCR5 as opposed to the human CCR5. So we mutated that single nucleotide to the rhesus form and then asked whether the shRNA still worked, keeping our fingers crossed that it would still work. And it turned out it did. The rhesus, if we use the rhesus, if we look at downregulation of rhesus CCR5, the rhesus form downregulates rhesus CCR5, but not human CCR5. And if we look at the human CCR5, the rhesus does not work on human, but the human does work on the human CCR5. So this tells us a couple things. First of all, we have a reagent that we can use in the monkeys, but secondly, it also helps establish the specificity of our assay system, of our shRNA. So we know that a single nucleotide can make it either functional or non-functional, depending on which species of CCR5 we're dealing with. So now we can take the rhesus shRNA and think about putting it into the monkey model in a transplant setting. So we transplanted 2 animals and 1 control animal. The two animals transplanted with the shRNA are shown here. The reconstitution with granulocytes, monocytes, and lymphocytes was fairly typical of other transplants that were done. Typically, within about 6 months, we begin to achieve stable levels of the transplanted, of the reconstituted cells. The real question was, are we getting shRNA expressed in the cells, and is it functional? So, if we take the lymphocytes from the monkeys and look at siRNA expression, we can in fact detect the siRNA within the lymphocytes of the animals. This band here is expression of the siRNA in the animals. And I won't bore you with the numbers here. And most importantly, though, is this shRNA against rhesus CCR5 functional in these transplanted animals? And the answer is yes. And we look at, here's one time point. This is a 5-month time point. These are the 2 animals, blood from the 2 animals transplanted with the shRNA against CCR5. This is a control animal here with vector alone. And you can see that, again, just like I showed you earlier with the in vitro experiments, if we look at the population of cells that's transduced, here, CCR5 is downregulated relative to the animal that is a control animal, as well as relative to those cells that are not transduced within each animal itself. Okay, so expression of shRNA in a transplant setting gives a stable CCR5 expression, and I can tell you that this is showing the expression of the showing the downregulation of CCR5 up to 9 months. We have the animals now are going on to about 2 years now, and they're continuing to show stable downregulation of CCR5 in the animals. As I said earlier, I know you all want to hear what the effects of this are on HIV-1 infection, and I can't tell you that because the animal experiments are really expensive. They cost about $40,000 to do a transplant per animal. And so we've only been able to do these 2 animals, and we want to continue to follow these 2 animals for a longer period of time to see if there's any adverse effects of having the shRNA in the animals, either effects due to the downregulation of CCR5, or, as I showed you earlier, perhaps more subtle toxic effects of just having the shRNA itself expressed within the animals. But I am confident that by using shRNA technology, and if you use the appropriate choose the appropriate shRNA, you can get downregulation of CCR5 in a transplant setting. And our hope is that in the future, we will be able to use this in human clinical trials where you would downregulate CCR5, say in an infected individual, and then those cells that are resistant to HIV-1 in that individual would then predominate over time. You may end up with an equilibrium between infected cells and non-infected cells, but with those cells that do not express CCR5, as Dr. O'Brien pointed out earlier, should have a selective advantage and predominate over the lifetime of the individual, hopefully preventing the development of AIDS, or at the very least slowing down the progression to AIDS. So, as acknowledgement, I want to acknowledge much of the initial work was done by Dr. Dong-Sung An in collaboration with Robert Donahue at NHLBI, who has a beautiful monkey facility, and in collaboration with Xiaofang Xin and David Baltimore at Caltech. And the more recent work on the macrophage transduction and expression of macrophages by Dr. Maza Kamada and Ming Liang in my group. Thank you very much. I'll be happy to take any questions. Steve.
Methodological question. This shRNA, does it replicate in dividing cells? How does it continue to regulate after several?
Because we have it's expressed from a lentiviral vector. So the vector is integrated into the cells and continues to express the shRNA, right? So, I mean, that's sorry if I didn't make that clear, that's a key point, because what we're aiming for is stable expression of the shRNA, hopefully over the lifetime of the monkeys or humans if we get to that point.
All right, well, I'm glad to see you're getting into this. It's something, obviously, that we thought about in all kinds of ways, trying to shut down CCR5 expression 10 years ago. But we were always worried that no matter what we did, we could never shut it all down, so it would never be a therapy that would prevent infection. But with respect to, it's interesting to see it with respect to infected individuals, because I agree with you, if you could diminish or outcompete, say, the HIV positive cells, it is possible that you might actually slow this very slow process.
Mm-hmm. That's our hope, and you're right. I mean, at this point, we're not able to completely shut down CCR5. I mean, if you look at the data, there is always some expression of CCR5 expression. Depending on the experiments, we're getting a knockdown of anywhere from 5 to 20-fold the levels of CCR5 within cells. So, but that might be enough to give those cells a selective advantage. And those cells, in theory, those cells that have the greater levels of knockdown should predominate over time, because they would have a selective advantage in infected individuals. At least that would be the hope.
Why does it cost $40,000 to do a monkey transplant?
You have to ask Robert Donahue that question. I mean, they have to do them. It's basically, the way he explains it to me is basically he has to create an ICU, for each transplanted animals, because they're fully myeloablated, and so they have to be kept under containment for some time after the transplant.
I want to get an estimate from the SPF cat people and FIV.
Then we have to see if the cat CCR5 is the same as the human. Yeah. But no, I think that what we want to do is we want to model this in chimeric mice. I mean, I think that's the direction we ultimately want to go. But we want to do a couple experiments in primates first, because again, I'd really like to see this happen in humans, and arguably the non-human primate model is the closest to that of humans. Crystal?
If they were totally myeloablated, you still saw a lot of CCR5 in their own endogenous cells. So why did those cells not disappear?
Maybe what referring to the cells that look like they're not transduced. So the cells that are not transduced, if they're myeloablated, the animals will die. If they're not transplanted, they'll die. So the cells that you see that are GFP negative, that's because we're not getting 100% of the stem cells transduced. So there's
Percentage was kind of low, though.
yeah. I mean, typically we achieve around, in a good transplant, we can get 12% transduced, marked with the vector. Majority of the cells still don't get transduced. Ben-Hur?
The obvious question is, do you plan to challenge that eventually? Since these are two precious.
Yeah, I think I mean, obviously we're itching to do that, but I just really, I think there's a lot of things that we can do first. I mean, we want to do some microarray analysis, which Don Song is planning to do, to see whether there's any differences between RNA expression profiles in transduced and non-transduced cells. Because even if there's no overt phenotypic effects of the shRNA expression, it would be worthwhile to look to see if there's any differences in RNA expression. Another thing we'd like to look at is do integration site analysis to see whether there might perhaps be any effects of preferential integration leading to malignancies or other types of phenotypes within the animals that we're not seeing grossly by just looking at bulk populations of cells. So these are some experiments I'd like to do first before we ultimately sacrifice the animals and challenge them. Questions? Thank you very much. And I think Gail had some closing remarks for us.
Thank you, Irvin. This has been a really exciting day for the AIDS Institute, for many of us in the leadership group who have come together to try to instill a lot of excitement in the research that's been ongoing at UCLA with regard to HIV and AIDS. So we were delighted that you took the time to spend the day with us to hear the presentations and participate in the discussions. We'd like for this to be the beginning of more dialogue between the university community and the AIDS Institute. It certainly is inspiring for investigators, it's hopefully inspiring for students, and it's very affirming for those faculty and investigators who are doing the work in innovative research areas that really need a lot of support and a lot of dialogue. Sometimes you think you're doing something good until you present it, and then you get another way of looking at things. So this has been great, and we hope that you enjoy the way that it was presented, and we hope that you enjoy the content. We'd like to be out of the box and be controversial a bit. I think this was a very challenging topic, and I think it was very well done by Dr. O'Brien and Dr. Fenton. So could we give them a hand of applause again? I'd also like to recognize not only Drs. Chen and Dr. Zach, our directors of the CFAR and the AIDS Institute, respectively, but also Dr. Mitsuyasu, Dr. Coates, I'm thinking of all the AIDS leadership group. Why am I blocking Mitsuyasu, Coates? And me. It didn't come to mind. For all of our work with regard to this day. However, had it not been for Rathna Natharajan Sweeney and for Gina Lee, who we're asking to stand because of their long overdue work and dedication to making this day possible. So really give them a rousing hand of applause. If you have other topics that you'd like to have us tackle, please take the time to write them down. I don't know that we have a formal evaluation of today, but let us know if you have some ideas that might come up that we could develop in the future. Finally, I want to share with you something very personal. This was an interesting week, I guess an interesting couple of weeks, in that we had the anniversary of some of the civil rights marches in Birmingham, Birmingham, Alabama. One of the little girls, Carolyn Robinson, was saluted at a organization activity that I went to this past weekend. This is one of the little girls who died in the bombings in Birmingham, due chiefly because of her race, because she was in a church at the right time, but at the hands of someone who didn't respect her need to grow, and she was not alone in the people who were affected by that incident. I've been old enough to see the effects of race in America. I grew up in a segregated country. We could not eat in restaurants or sleep in hotels, and my parents did their best to let us know that race was not an important part of who we were, that we were the most important parts of their lives because of the character of my sister and me. But I want you to know that it's never been a thought that never left my mind that race wasn't important because of the way that I was treated. So I'm very proud that we address this issue today. We shouldn't be afraid of issues. It's how we handle it, isn't it? It's not the topic themselves. For me to get to the point where I could see that being categorized on my birth certificate as a little colored girl, to an African-descended woman to have a better understanding of my genes and yet the health disparities means a great deal. I hope that each of you will personalize how important it is for us to tackle the things that other people are afraid to discuss. It's not politically correctness we want to achieve, it's scientific accuracy. If we do that, we can debunk a lot of myths, we can give people the facts, and we can be one people. Thank you so much for coming today.